That should do the trick. Sorry about the delay. Welcome, everyone, to Neuroccino on the 16th of November. I'm aware of 2 to 3 papers today. Can I just... Well done. Perfectly trained. They're all in the chat. Thank you for that. Okay, with that, let's just not lose more time and crack on. Laura, over to you. Just share your screen.
Okay. I'll try to present this paper this morning, which came out a couple of days ago in Scientific Reports. It is a proposal for a new computational framework for the investigation of the shape and the profile of brain pathways across different populations. And it is called bundle analytics. So it is a tool that is freely available in the IPI. As many technical papers do, there is a first part in which the technical part is outlined, and then in a second part, the authors tested their tool on a data set, so we can see both of them. I'll start with the technical part. So there are different steps for this bundle analytic tool to work. The first step, which is this A here, consists in extracting the bundles from a whole brain tractogram. So what you need to give as an input to the program is a whole brain tractogram, and you also need to give it model, an atlas model of the bundles of the white matter pathways of the brain. I can anticipate what they did in the study that they did to test this tool. They basically use an atlas of white matter pathways, which was coming from the Human Connectome project. Then what the program does is to apply a streamline-based linear registration so that the whole brain tractogram is registered on the model atlas. And here we have it. Then for the second step, we use this product of the first computation together with a model of the bundle that we want to extract from this whole brain tractogram. And then RICO-bundles allows to extract the bundle of interest from the whole brain tractogram that we have. So the authors also propose that a further step can be applied to RICO-bundles, which they call calibration. So that from what I got, basically you use the extracted bundle as a model bundle, and then you rerun a second step of RICO-bundles. This is due to the fact that we are using an atlas for extracting a population-based atlas for extracting the bundle in the population. And then in order to have a more anatomically plausible version of the bundle, they use this type of trick. They also say that it's very efficient when you need to extract bundles that are very small or that are different to disentangle in the whole brain tractogram. Then there is this second step here, which consists in the segmentation of the bundle of interest. So, okay, they call it the assignment step, because for each point in the bundle of interest, they assign the closest point in the model of the bundle of interest. And basically they plot the centroids of the clusters that are extracted from the model bundle upon the bundle that we extract. So what we get, it's a segmentation along the length of the bundle of interest into different small segments. So the first step is the tractometric analysis, meaning that we use the bundles that we segmented in the two populations. So here we have two groups. For example, the red one is the patient one, and the green one is the healthy controls one. We have the reconstruction and the segmentation of the bundle in all the subjects for the two groups. And then an analysis to see whether the two groups significantly differ in some segments of the bundle of interest, which is done through a linear mixed model. What we get, it's these type of graphs, so that here you can see we have two different thresholds of significances, which are p less than 0.01 or p less than 0.001. They consider as significant the higher, let's say the lower threshold, so the higher significance. And you can see these are the segments that we that were defined upon the bundle of interest. And the sequence of the segments which are considered significantly different between the two groups, you can see it here. 4 different measures, which are, for example, fractional anisotropy, mean diffusivity, axial diffusivity. The nice thing about the tool is that then you can visualize on the bundle of interest the tracks, the segments of the bundle in which the two populations different. For example, here we see that the two populations differ in mean diffusivity and axial diffusivity in this particular segment, and for 2 measures of fractional anisotropy, they differ in this segment of the bundle. Okay. Then there is a last step, which is the comparison of the shapes of the bundles extracted between 2 different populations. Again, we have the, let's say, patients population, for example, and the healthy controls. In this case, we are not inputting the reconstruction of the bundle that is segmented, but the normal one without the segmentation. And they use this measure, which they call bundle adjacency. I will try to explain this to you, but I'm not sure I will manage to. So let's see. Wait. We have an example here. So basically, what they do is to compare 2 bundles. In this case, they consider this reconstruction of the arcuate fasciculus in the left hemisphere, and they try to test this bundle adjacency analysis with the bundle itself. Here, in C1, it's the copy of the bundle that we are considering. Then we have different subcomponents of the bundle that we are considering. So we see that we use different threshold, which is theta, equal to 5. It's a more strict threshold, let's say, and then a more broad threshold, which is 15. We have the results for the bundle adjacency score, which can range from 0 to 1, with 1 meaning that we have a complete overlapping between the 2 bundles in terms of shape similarity, and 0 when we have basically no line which is the same between the 2 bundles that we are considering. So we have the different thresholds, the different scores. You can see that they vary based on the threshold that we decide to apply. And then what you can have with this analysis, it's a graph of that connects all the bundles that we compare. So let's say in a whole population, you can see how the bundle differ between each subject and the degree of their similarity. Since let's say the group that we analyze can be very broad, then the best way to represent this similarity is with a similarity matrix that you can see here. So the complete overlap between the 2 bundles that we are analyzing is represented in dark blue, and then we shade to almost white when we have no similarity between the 2 bundles. Okay, so going back to what we were saying before, okay, we see that, sorry, this is what they did. So they computed, you can compute a similarity matrix, and then you can see which is the relationship between the 2 bundles of interest. Okay, we can move to the part in which they try to test this tool on a set of data. In this respect, they used this dataset, which is Parkinson Progression Markers Initiative. So they considered 64 subjects, 32 controls, and 32 patients. What they did, according to the first step and the second step of the tool, let's say, they extracted the bundle of the whole brain tractograms of the subjects, of the participants, and then they segmented these bundles with the assignment step, as I call them. Here we can see the results for this analysis for the fractional anisotropy measure. I forgot to say that what they did is to consider 30 white matter pathways of the human brain. So for each of the bundle that they consider, we have a plot. We can see that right are opposed and plotted with the different colors. And some of them show a significant difference between the two populations when considering particular sections of the bundle. For example, we can see here inferior longitudinal fasciculus, the two groups differ significantly in this range of sequences. For example, I don't know, it would be 75 to 78 kind of. Then we have the same for other tracts, such as the uncinate fasciculus, in this case in the left hemisphere, which differ between the two populations in this range of sequences. The overall results are presented here. For all the measures that they consider, we have some anisotropy measures and we have some diffusivity measures. And overall, what we can see is that for what concerns the anisotropy measures, so fractional anisotropy, constant solid angle, generalized fractional anisotropy, and quantitative anisotropy, overall, by comparing the two groups, when we have a difference, we have higher scores for the patients, meaning that when we have a difference, usually patients for the fractional anisotropy measures have higher fractional anisotropy. On the other hand, when we consider the diffusivity measures, the patients usually have the opposite pattern, so lower scores. This is the example of the inferior fronto-occipital fasciculus, and I don't know, we can go through this. So basically, you see that they extracted the bundle, they segmented it as before, and then here we have the plots of the differences between the two groups. We see that for what concerns the fractional anisotropy, the two groups differ in this region, which would be, I don't know, 58 to 60. And then for the generalized fractional anisotropy, we have the same. So overall, the results say that we have some significant difference in the different measures in this range of sequence, in this area of the bundle. So they also analyzed the entity of this difference. And when here you can see, this is the cumulative number of this is the number of the participants. And this is the value of the fractional anisotropy or diffusivity or whatever. And basically, patients in red show to have, on average, higher fractional anisotropy values when compared to controls in this range of let's say, in these segments of the IFOF. This is true also for the other two anisotropy measures. And this is not true for the radial diffusivity, in which usually control have higher scores. Okay. So then they computed, as said before, the similarity matrices. And basically, you can see that there are darker matrices, meaning that there is no great variability of the features of the bundles between participants. They also did this hierarchical clustering in order to see whether there were significant differences between the two groups, the healthy and the patients group. Let's say that they don't discuss this type of results this much. because it's not the main goal of the study. What they did is also to carry out an analysis of the performance of this tool compared to another tool, which is automated fiber tract quantification. But I don't know this tool, so if you, I don't know, if you have any idea on how this works and you want to discuss it, we can do it. And that's all. If you have any question, I'll try to answer.
Thank you very much. Are there questions in the room?
Can I?
Yeah, sure. Go ahead, Lia.
So thank you very much. It was really interesting. I was wondering, since they did a so detailed comparison for microstructural measures and also shape of the bundles, if it could be also interesting to compare like the right arcuate with the left arcuate, so hemispheric asymmetries in healthy controls using this tool. They say?
I mean, they don't do it and they don't mention it. But I guess I think it's something feasible. I don't know if you guys have an opinion on this.
No, because when I saw the figure of the IFOF, I saw like the different terminations, the cortical terminations. So I was thinking that they could be used. There are plenty of possibility to use this tool. It's really interesting.
Yeah, I agree with you. Yeah.
Any other questions? Leonardo is looking more and more professional every week. In that case, I have two questions for you. The first one is that you, in the beginning, said that they used an atlas to dissect it, and I was wondering how limited the method is depending on which atlas you use.
Yeah, of course, you bias the extraction of your components based on the atlas that you use. But I think that one way in which they try to walk around this situation is to use that last step in RICO bundles. So to use the track that you extract and as a model bundle, and then to rerun the RICO bundle process. In some way trying to, I don't know, to bridge for this difference.
So again, the results are only as good as the atlas that you use. The second question I had for you is the figure you showed with the IFOF and the little segment that was highlighted to be different between the two groups. When I saw that, I was wondering what the anatomical relevance actually is of knowing that a little stretch of the tract is different rather than its connection or degeneration along the pathway. Yeah, there you can see it. So to me, that's like, it's too clear cut, literally, to be anatomically or clinically relevant.
I don't know, but I guess if these, for example, there is always in patients compared to control, there is a systematical difference between healthy and patients in this particular section for some measures such as fractional anisotropy and stuff. It could mean something. I don't have the answer, but
sorry, I didn't expect you to have the answer. It's just I saw it and that
sure.
It's more a comment than a question, I guess.
I don't know.
Thank you. Okay.
I also
Any
have
other
a small question.
Yeah.
How can they be sure that they are comparing always the same segment in different subjects? Because if two people have different size of the brain, I mean, let's say, of course, the track will have different length. Therefore, the element 50, well, no, the one in the middle actually is the only one which is okay, but possibly. But let's say, yeah, 30% of the bundle. How can you be sure that you're looking at the same part of the bundle in two subjects which have a different bundle length.
Yeah, they wrap everything in the MNI space. So
okay,
this might
because the assumption is when you put things in the MNI space, everybody is the same. But that's an assumption.
Yeah.
But is it actually correct in your experience? I mean, does it actually?
It's not correct. It's an assumption.
Okay.
It's like, you know, if you want to move on, you just assume that it, you know, it's correct. But if you start looking at inter-individual viability, you realize that you can't really stretch brains that easy.
Okay, are there any other questions? No one is unmuting or raising their hand as far as I can see. Okay, in that case, I'm going to throw in a paper very quickly that I found this week and thought we could all do with some positive news these days. Hang on. Multitasking across two computers. Just need to find where it is. Share screen. Here we go. Okay. Now the advantage is I don't need you to ask if you can see my screen because I know you do. Okay, so this is a bit of brain topic, which is totally legit. I get asked that question every week if people can present papers that aren't strictly neuroscientific. And yes, you can. It's about exciting science that attracts your attention. So I saw this headline, satisfaction of scientists during the COVID-19 pandemic lockdown. I'm like, I have a subjective feeling. I know what Twitter is feeling. But it was interesting to look at the numbers. So this is a study from Switzerland, and they were fortunate enough to look at satisfaction with work-life balance, work, private life, just before the pandemic started. So they just kept it rolling and collected data pre and post lockdown. Very fortunate timing. And they collected data from scientists in German-speaking countries, so Austria, Germany, and Switzerland, across all levels from PhD to professorial level. In total, they get 13,000 and a bit responses. And overall, they found that scientists are quite happy with lockdown. The p-values are high, the effect sizes are not as high. But I thought it's good news, B, it's quite interesting to actually stick numbers to these events, and it's quite interesting that they just happen to collect the data at the right time in time. So they make a couple of statements why that might be the case, and also postulate that they can generalize the results to the more wider public, some of which I do agree and some of which I disagree with personally. But overall, the assumption is that life as a scientist, we're used to dealing with complicated data, but also we are quite used to uncertainty and dealing with uncertainty. But also our jobs are, again, that's country dependent for the countries they're talking about, are relatively safe at the moment, which obviously having job security and not having to worry about economic losses helps. They also, so here you can see they collected data from February to April. As I said, very fortunate timing. They did a before and after comparison with their t-test, and then also fed it into a regression analysis. You can see here the development of the data over time. This grayed out area in the middle is when lockdown began in those three countries. So there's a range of a weekend, basically. Now, for me, interestingly, there already seemed to have been a slightly positive upwards trend even before. And the other interesting thing, I guess, is the impact of kids, having kids at home or not having kids at home, especially during lockdown 1, where most schools were shut. And they found that while the satisfaction overall slightly increased in scientists, it was more so in scientists without kids at home. And the satisfaction of scientists with children did not change significantly, if I remember correctly. Yes.
It makes sense. It makes sense.
I was thinking of you when I read it. So as I said, you can see that the effect sizes aren't too large, even though we have a large p-value. And I know this nicely links in with the next paper that's coming, so it's nice to have them as a combo. But overall, I thought it's time for good news, and we're clearly a good bunch of working group to deal with this for the time being, also because we are used to working from home, I guess. Happy to take questions if you have any. Anyone?
Ma'am, one question.
Yeah.
Yeah. Does the study involve the satisfaction of the scientist based on the right decision made by the government? I could see the personal question, like children and all sorts of things. But this scientist were scientists also asked about whether the decision of lockdown was right or wrong. On that basis, they answered.
Yeah, so I forgot to mention, this is obviously an online survey where people just comment like subjectively. There is a whole section in the discussion that dealt with the UK. Personally, I was a bit hesitant to go through it because I find it quite painful. But obviously that is, it's limited to the 3 countries, Austria, Switzerland, and Germany. And I haven't actually seen the list of questions as such. They may be in the supplementary material. So I don't know if they did inquire about your happiness with your government. It wasn't a big point in the paper for sure until the discussion about the difference with the UK, but it's also a different, well, it's not only different governmental decisions, it's also a different academic system and funding system. So there's more things to consider than just.
Yeah, what I thought is, in general, scientists would be happy that the lockdown did take place because of the control over the coronavirus cases, right? So that could be one of the factors, but in general, right?
I think there's a lot more you could look at. Bear in mind, they didn't actually plan the study, it just happened to coincide with the lockdown. Right. But personally, I thought it was interesting to stick a couple of numbers to what has happened. And I gave an interview a while ago about the impact of Corona on our scientific lives. And I've raised a couple of points that at part, personally, I feel more satisfied with my work-life balance at the moment. So it was quite interesting to me personally to see that it also has a positive impact for some people. And generally just having good news these days just made me happy. That's why I thought I'd throw it in. Right, but now over to Michel, who will tell us more about the importance of the effect size.
Thank you, Steffi. I'm gonna share my screen. I I can find the paper. Good thing if I find the paper. There you go. And then, sorry for this. Just gonna share my screen then. Ah, no, this must be the one. Ah, can you all see my screen? Great. So, all right. So it's a methodologically complex paper, which I'm gonna be honest with you, I didn't really understand the methods. But this is a good example how you can read a paper, you don't understand the methods, but still extract important information that you can use. So full disclosure, I won't be able to explain to you how they did it. But in general, as we've seen with the papers that Steffi reported, if you do a comparison of on large samples of subjects, you're very likely to find something significant. And this is how the authors of this paper start their paper. This is like, for example, they look at online dating, and they tested 19,000 married Americans, and they check whether there was a relationship between the fact that they met on online dating or not, and whether there is an effect on their happiness in their couple and whether they stay together or not, and find a very significant effect that made the press, and everybody was very impressed that online dating was so much helping our social life and love life. However, what they stress is that because they tested on so many people, it was highly significant, but there was a very, very small effect size, which means that compared to people that met not using online dating, people who met using online dating, if you were taking 100 couples, only one more divorced for those who didn't meet online. And so that's like a very small chance or small improvement compared to meeting classically in everyday life. However, because the p-value and the statistical significance was so big, it's been inflated by the press. It's been inflated in the mind of people that think it is so much better. And so there is a kind of mix up between an effect being significant and the size of this effect in the literature. And this is especially true now that we have large databases such as the Human Connectome Project, which is more than 1,000 subjects, or the UK Biobank, which is more than 10,000 subjects. So what they bring in there is bring back the importance of calculating effect size when you're doing statistics, especially on large populations. But they also stress another problem, which is specific to fMRI and VBM sometimes, which is we're doing correction for multiple comparisons. The more tests you do, the more you have to correct your p-value, because if you do 1 million tests, you're going to find something significant just by chance, and you need to correct for that. So in fMRI, researcher kind of tweaks the thing. They do a correction on the number of clusters that are significant, that are contiguous voxels together, that will show some significance. But there, by doing so, they're also doing like some spatial resolution. So you know your cluster is significant, but you don't know really where inside that cluster. And that's another issue with the way we're doing neuroimaging. So they propose a way to solve that, because the paper is not about highlighting all the problem we have when we're doing research on neuroimaging. It's also about proposing a new way to assess this, which is a confidence set for Cohen-D effect size, which combine the confidence interval, effect size of Cohen-D, which tell you how much of the difference is explained by the effect that you've been reporting as significant, with the spatial certainty that your result is very well localized inside this cluster. And so they apply it to the human connectome project, if I remember well. That's gonna happen. There you go. And so here you have typical activations that are related to working memory. And they're all significant. Where you have colors, that is all significant. But if you apply this confidence set for coherence D, you're able to disentangle the voxel in blue, where it's significant, but there is not a very large effect size. It's like very small effect size, and you're not sure at all where it is in these big clusters. Yellow, which is quite satisfying, where you're like you have an idea, like it is significant with an effect size around 0.5, which is a large effect size, but just on the limit. And you have a kind of an idea that it is well located inside your cluster. And the red one, which is like the area where you have most of the effect and the difference that is related to the function that you're studying. And this is at the group level. So you can imagine that a lot of effect size is lost due to inter-individual variability. Okay. Then, so that's for the part I understood. Then you get all of these equations over here. See, I have no idea. They did like some simulation over there with more equations. Da-da-dee-da-da, more. It looks really great. It's just, it's not, it's not really my field of expertise, so I cannot deal with it. But, if somebody want to jump in and understand what's going on, just tell me. It's magnific, beautiful, like quantification of the noise and simulations of this effect. See, it's a distribution of effect size for the UK Biobank, which is nicely centered on a negative value. It's not, I guess, not that really good. And yeah, that's pretty much it. I'll be happy to take questions.
Lovely, thank you, Michel. I'm glad you didn't get any further than I did in this paper. Makes me feel really good.
I want to disclose that Steffi sent me this paper yesterday. And I think like the message that is important to really know and learn is that effect size is actually the real true value that you are interested with if you want to do good science. P-value is just, it's, it doesn't mean anything. It's going to be more significant as you increase your sample size.
Are there any questions?
Can I ask, are they suggest any tools or
yes,
just...
Thank you for this question, Lia. At the end of the paper, they actually report that they wrote the code. I remember where it is, right here. Data availability, all code used for the simulation and the analysis of the HCP data are available as open code that you can take and apply to your own data if you would like.
Okay, great.
Yes, testing it right now. Are there other questions? Yes, Leonardo.
I would just like to point out that it's true confidence intervals effect size is extremely more important than the p-values when we have very big numbers. We have also to remember that we have when we have small numbers like 20 participants or so, the estimation of the effect size can be actually inflated. So actually, when we are on the other side of the spectrum, looking only at the effect size can be misleading because we can estimate a large effect size on our small group that cannot be replicated on a different group.
But will the p-value be significant as well?
Absolutely, yes.
Yeah.
Absolutely. As a matter of fact, I mean, you can have larger effects for less, so to say, less. Yeah. One should never say less significant, but we know what we mean for less significant p-values with small samples than with, and the opposite with big samples.
I think we had like a little segment like a month ago at Neuroccino where we presented this paper from your former colleague or current colleague in you know, where you're working, where he's like pushing forward Bayesian statistics that was in Nature Neuroscience and saying that that was covering very much for this kind of problems.
It's a very nice paper indeed. As a matter of fact, there is also a video tutorial on that. It was not exactly on that topic. Actually, the main topic of that paper is the fact that when we have a p-value which we deem as not significant, we don't have an evidence of the fact that it's not significant. Correct. I mean, we don't have any evidence. I mean, we cannot say anything about how significant is the fact of not having an effect. Sorry, when we're dealing with the frequentist terms, is always a mess. But yeah, basically that's the idea. When we fail to reject the null hypothesis, we do not have any measure of how confident we are in rejecting the null hypothesis. And in that case, Bayesian statistics can help because it can tell you something about the confidence with which you reject the null hypothesis. I mean, to say it in human terms, one big problem, of course, also for MRI images is the fact that, as you said, we have lots of voxels. Therefore, if we apply very strict corrections, we get rid of false positives, but then we might have a lot of false negatives. And now the question is, okay, I have this region that, according to which the comparison for multiple correction tells me that I should not trust that there is any effect there. But how confident can I be in the fact that this is a true negative instead of a false negative? Okay, so it's a slightly different thing, but it's in the same, it deals with the same problem, so to say. Did you already discuss this paper before?
Yes, we did discuss this paper months ago. I actually presented it.
Ah, okay.
I'll send you the link.
Well, I already read the preprint before
no,
it was published.
So but, you can tell me if I did all right.
But
yeah, but just to let you know that actually this kind of calculation are very easily carried out in JASP. JASP is a statistical tool, very well done based on our code, which is developed here at the University of Amsterdam. And they implement a lot of Bayesian version of typical statistical tests.
It's great.
In a nice way, in a sense that it's very easy to use.
I did promote JASP, and it's great, but not for very large matrices.
Unfortunately not. Yeah, indeed.
All right, cool. Thanks.
Lovely. Thank you both for that discussion. Very interesting. We have one more paper from Lia, and we got about 10 minutes left. So I just hand over to you straight.
Okay. Share. Right, it's the screen, I guess. Yes. Looking to the published paper last week, I went through this one. Is a Nature review about primary diseases, and it's about delirium. And it was really interested both for the biochemical perspective that try to explain the pathological mechanism underneath this clinical presentation of delirium. And also, I didn't know that it was a so common condition that we can have in elderly patients and also healthy controls, and in patients after surgery. Indeed, there are a lot of comorbidity factors that can trigger this condition. That can be dementia, age, drugs, depression, and also post-operative conditions, in particular in patients in intensive care and other ventilated and general hospital condition. The hypotheses that trigger delirium are poor understood, and one of the main explanation is according to the metabolic dysfunction. Here in this nice picture, you have represented neurons and an neuron and an astrocytes. The function of the astrocytes is to provide help neurons in the metabolic pathway. In particular, you have this lactose shunt, and here the mitochondrial ATP synthesization. And in some diseases, like neurodegeneration, you have dysfunction of these glucose transporters. Indeed, the nutrients to the neurons and astrocytes are brought through the blood vessel barrier. You have oxygen, glucose, and so if you have a comorbidity of neurodegeneration and you have a neurometabolic imbalance, that could trigger delirium. Also, another possible trigger is inflammatory mechanisms. Indeed, here we have the barrier, the periphery, and the brain tissues. If you have an reaction, so immunic reaction of the systemic inflammation, you have leukocytes, macrophages that go inside the brain barrier, the parenchyma, and trigger mechanism to the macroglia, astrocytes, neurons, to produce some chemical species like reactive oxygen species, reactive nitrogen species that interact with this neurological and metabolic balance, and this could trigger delirium and also accelerated dementia mechanisms. And also here you have the combination of the macroglia, astrocytes, and neurons with the neurotransmitter system, in particular the arousal system that can interact and create an imbalance and also trigger delirium. And also here it was interesting because they talk about the current pandemic and sources, and so the COVID-19 problem, disease, and in particular they say that due to this condition, the pathways that we have for treat delirium that are standardized are not, here we have some examples, are not well followed because we are in emergency, we have a problem in intense care units, and so now the most common treatment, like in the about 60% of patients, is sedation. But also, if we look to the treatment using antibiotics, we don't see a great improvement. Sorry for scroll down, is here. So placebo, antipsychotic drugs, we don't have a great change in the clinical conditions in these patients. So also this is debated. And according to neuroimaging, they use CT and MRI to look into perfusion or also you can use PET FDG to see the glucose uptake, or also MRI spectroscopy to see the lactose level, and so to in vivo measure the metabolic imbalance. And just to give an idea, where here also they discuss some neuroimaging findings. So there are studies in preclinical studies, so animal models, and also in patients in acute conditions, or also chronic patients presented dementia that underwent fMRI and PET scans. So according to PET scans, you see that there is a specific pattern of glucose uptake in dementia patients who present delirium, and also according to fMRI and EEG measures, we have a different relationship between the dorsolateral prefrontal cortex that is involved in executive functions and the posterior cingulate cortex, who is part of the default mode network. Usually they are negatively correlated, and in the presence of delirium, this relationship is inverted. And also using graph measures in fMRI and EEG, functional connectivity matrices, you have variations of the graph measures, so reduced connectivity strength, reduced global efficiency, and reduce the local clustering and modularity. So a different functional connectivity according to these conditions. And I wanted to point out some numbers, because this is a common medical emergency. You have about of 25% of older medical patients that present this delirium manifestation, and around half of patients in intensive care unit, and at least 1 in 7 of hospitalized patients overall. So this is a common disease that I think is important to talk about. And yes, here they explain what is happening here in the current pandemic. Another thing is the quality of life, because also if you have this acute illness, so delirium, as we have seen, there are effects on the brain functioning, metabolically speaking, and actually we don't know what really happening. So these can trigger cognitive impairment, depression, cognitive disability, and so lead to a poor quality of life also after the delirium manifestation, so in a long-term perspective. And I will jump to the outlines. Here they say that there is so like one size fits all. So the delirium characterization is too general to understand what is going on. So like in other diseases, like mood disorders, dementia, we need to to categorize the delirium phenotypes to also understand which can be the treatment, because as they shown, the psychotic treatment is not seems not to be an evidence that is a good treatment. Or for example, one categorization that they are starting to do is according to motor activity during delirium, so hyperactive, or mixed. And so the suggestion is to split the delirium into subtypes to better understand the pathophysiology underneath, and also the like imaging that you can have specifically for some conditions or not. And yes, they say that there is this extraordinary gap in the treatment of these patients, since they show, and there are a lot of reviews, Cochrane reviews about this disease that is so common. So it's, I will stop sharing. I try to be fast.
Thank you, Lia. Very interesting. Are there any questions? No question at the moment. Okay, great. Personally, I was quite surprised to see how common delirium is. Yeah. 1 in 4 or 50% on ICS. That's a lot of people that are affected by it. Thank you very much for that. If there is no questions, no. Thank you for joining today. Just to highlight, we had the CNS Comms event last week. So if you missed it, it's on the YouTube channel. You can catch up 10 talks on structural connectivity of the cerebral cortex. And I guess we see you next week. Thank you all for coming. Bye.
Thanks a lot. Bye.
Bye.
Bye.